So I'm in great company this morning with these three panelists, and so let's get started with some introductions. So I guess we'll start with Andy to my left, and Andy is Principal Technical Development Lead for Genentech, but he has a long history in cell and gene therapy, and I think you started with cell genesis. So Andy, please.
Yeah, I'm Andy Lin, happy to be here. I'm a technical development team leader. What that means for Genentech is I lead CMC teams from early stages all the way to launching products. So my most recent product that I helped launch was Polivy. It's an antibody-drug conjugate that was launched a couple years ago in B-cell lymphoma. And so that was a great pleasure to work on that program. I've been at Genentech for about 13 years altogether. As you may know, Genentech recently signed a deal with Adaptimmune to develop iPSC-derived T-cell products, so the allogeneic-based product. We also have a collaboration with BioNTech, developing mRNA. We also have quite a bit of gene therapy exposure these days with notably Spark acquisition a few years back. We also are working with adapted biotechnology in developing T-cell therapy based on their TCR discovery engine. So Genentech, although not well known for cell therapy, but my person next to me has a lot more experience in that area. We are dipping in that. And previously, I was with a company called Cell Genesis that did a lot of early, before it became really popular, like today, in cell and gene therapy, worked with Carl June years ago, developing the first CAR T for HIV, of all things, and he, of course, took it. That became Kymriah today. So had a really great privilege working with him in the early days. We call him using Dynabeads years ago. Had a big magnet that we used to separate cells with. It's still in use, which is amazing. So it's great to see the field has really, and we'll talk about industrialization today based on our experience in other areas and how that's so critical for this field. Yeah, so.
Great. So next we have Sophia Sharp Donaldson. She joins us from Kite Gilead, where she serves as the Executive Director for Strategic Sourcing, Procurement, Supply Chain. Anything else? Please.
Good morning, everyone. As Betty has shared, I'm Sophia Sharp Donaldson, and I work for Kite Pharma, which is a company of Gilead. And we are one of the few companies, I'm happy to say right now, but others hopefully will join us, that have 2 commercial products on the marketplace in the CAR T space. And we are actually about 3,000 employees. We're headquartered in Santa Monica, California. Our parent company, Gilead, is headquartered in San Francisco, and we have about 5 manufacturing facilities around the world, with 2 of them coming on board in the next year or 2. We have 2 based in California, 2 based in California with the new viral vector coming online soon in California. We have a Maryland facility as well coming online next year, and we have a Netherlands facility. So we're really proud and happy about that. And again, we're focused on CAR Ts, autologous, right now in the malignancies of hematologics. The large B-cell lymphomas is what we focus on today. And we are partnered with Appia and Shoreline to help us bring forward our allogeneic platforms. So we're just entered into some relationships with them as well. So I'm really happy to be here. My primary role on this panel is just to provide you a supply chain perspective of this industrialization that we have going on. It's an intricate part because especially in the CAR T space, it's really make to patient orders. And so your supply chain becomes very integrated into your manufacturing processes in order for us to help patients that are waiting. So I'm really happy to be here and with this esteemed panel today.
Finally, we have Jerry Kasia, who is bringing us a biotech innovator point of view. Jerry currently serves as Chief Technology Officer for Graphite Bio.
Yeah, well, thanks, Betty. And I have to say, you undersold. I wasn't sure after the networking bash.
Undersold?
You undersold the attendance here. I think it's a pretty darn good attendance. So I commend you all for making it after the networking bash. You'll know just to know your panel a little better. I think we all had a quiet dinner last night, so we could get to know each other even better. And we realized that we're all morning people, so that's a good thing, I think. I don't know that, Betty, I don't know that you planned that, but it's a good side effect. So anyways, yeah, so welcome everyone. I'm Jerry Cation. I'm here as part of Graphite Bio, which I joined in April 1st of this year. And my career prior to that was 32 years at Genentech and Roche in a number of different roles, basically around the CMC table, let's say. And so I was really excited to have the conversation today and learn from the panel, learn from all of you in terms of what industrialization looks like. And of course, sometimes we have to look back to go forward. And certainly as I was reflecting on participating in this panel, the 32 years in biotech, many of the same challenges, as you guys know, were present 30 years ago, open questions, which have since been, I'll call it, solved in terms of industrializing platforms in biologics, and of course our challenge is to do that with cell and gene therapies. The company I'm with now, Graphite Bio, was formed in April of 2020, so basically formed during a pandemic, and it's a next generation gene editing company which is born out of Stanford University and under the founders from Stanford, Matt Porteous, and the work from Matt Porteous's lab, as well as Maria Grazia Roncarolo. And so we're preparing to enroll our first patient. We will have our first patient enrolled. We're on track to have our first patient enrolled for our sickle cell treatment by the end of the year. And that's a pretty fast ride if you think about forming a company in April of last year to enrolling your first patient. And if you ask the question, gee, how does a company move so fast? It's really based on a long history of gene editing research that Drs. Porteous and Roncarolo have done throughout their careers. So it's a real pleasure to be there. And you might ask too, why are they? Why would a company be looking for someone to help build the CMC capabilities? And really, it's because our programs, as you guys know, can move very, very quickly through the clinic. And so I'm currently building a team. We're a company of 60 people right now. I joined on April 1st of this year as employee number 29, and so we're in that growth phase. And our doubling times are still pretty short right now, but we're really looking forward to seeing what kind of contributions we can make to the field.
Thanks, everybody. So I will serve as your moderator today. My name is Betty Wu. I am with Thermo Fisher Scientific for almost 18 years now. More recently came into a role as Vice President and General Manager of our cell and gene therapy business. So again, why don't we get started with the session. So I'll keep my comments brief so that we can take full advantage of the knowledge and experience of this panel as we kind of navigate through some of the challenges and opportunities with industrializing cell therapy, possibly drawing on some learnings from more traditional biologics manufacturing. Some principles will extend, others will not, so we'll tease that out today. So one of the themes that came through loud and clear from day one was the need to scale. And certainly one of the levers we can pull here is automation, right? So through automation, we hope to address process efficiency, robustness, safety, quality, costs, so many benefits there. A second theme that we're going to be centering on today, as Sophia had mentioned, is going to be around supply chain. So I certainly think the learnings from the pandemic forced us to be more efficient and more thoughtful in the way we approach supply chain issues and opportunities. So we're looking to Sophia for her expertise there. A third theme that we'll touch on today is around the broad topic of standardization, but I think we're going to dive a little bit more deeply into standardization as applied to analytics. So this is in-process analytics, QC analytics, so I think there's definitely some lessons that we can learn from biologics there. And then finally, we're going to start with the topic of innovation in fit-for-purpose closed manufacturing systems. I think we've reached an inflection point where we need to move from small-scale open workflows into closed, more efficient, more scalable, automated flows. And so with that, I think I'm going to start the questioning with Andy. So Andy, what are the main challenges in cell therapy manufacturing that are not addressable by solutions available today for biologics?
Yeah, I think in the biologics area, we've done quite a bit of work to standardize, right? We have platform processes, and one example is how quickly, for example, we were able to scale up the COVID-19 antibody production between Genentech and Regeneron, right? And that was done within a very short time, and the reason for that is platform, right? And I think that's, if I look at the cell therapy space, you have different vendors, different equipment, different processes, different reagents, and it's not that anyone has the best solution, but I think that is something that one can look to the biologics area, where we spent many years standardizing the platform. So I think that's not there yet for cell therapy. It's a good place to get to. So I think that's something that comes to my mind.
So maybe we'll kind of riff off of that theme a little bit. Sophia, any comments you want to make around critical factors that we may need to overcome in order to scale autologous cell therapy to a level that it might become mainstream at some point in time in the future?
Yeah, I'll focus more on something that is often not thought about, which is the single-use technologies that are in place today. Many of our partners and suppliers, as we know, they come with their technology, and it also comes with consumables. And often they can't be decoupled per se, so you're sort of working with a partner around their solution in its entirety. And some of those components and elements of that are made by medical device providers, and they may not be as advanced in aseptic manufacturing, and so you often see a gap in what's needed in the cell and gene therapy space, specifically in the cell therapy space, when it comes to meeting requirements like Pharmacopoeia USP 790, for example, where we absolutely cannot have particulates in our products. And because you've got sub-tier suppliers that are supporting these suppliers that are making our single-use technologies, they're often following medical device regulations. And so there's a gap. And I think often in this space, specifically in clinical manufacturing, it may not be as much of a gap, but when you move to commercialization, those standards and requirements from regs become very, very important, and they make the difference in your success rate in manufacturing reliably. So it's an area that is not often thought about, but I think it's something that we've got the platforms, but we certainly need to look at the single-use technologies and their sub-tier suppliers and the requirements we need at the aseptic, the ISO standards that we need in cell therapy, which is quite different, and advancing from the biologics space of antibodies or MABs.
Jerry, any other comments you might want to make in terms of what we need to do in order to get cell therapies to be the front line?
Yeah, I mean, as Sophia was saying, it's great. First of all, I have to say that it's my first time at this conference. I think it's all of our first time here. And I don't know, I talked to a few people that have been coming to this conference since it first started, and what's really great about this venue is that we get the opportunity to interact directly with our suppliers and express our needs directly to application scientists and things that can really help us solve problems, real world problems. And so to your point, Sophia, I think there are, as you point out, at the end of the day, we're making medicines that are being administered to patients, and with that comes the responsibility around aseptic processing and assuring we have sterile products. And yeah, that's a pretty high bar, even for an established industry like where we've industrialized, like the biologics industry. You still see problems that companies run into out there, and so we very much need to be working with our vendors and suppliers to I mean, I think in our program where we kind of talked about it as a closed loop process, but it's even more than that. It's making sure that we understand, it can be closed to begin with, but how do we prevent things like ingress, leaks? How do we assure that, the supply chains for materials, media, and things like that themselves are produced in a way that's going to be, as you pointed out, Sophia, going to be, meet our expectations for GMP standards. So I have to say, just being here at this venue and having the opportunity to see some of the companies that are now really addressing these problems head on, and particular suppliers, is really great, and we should all take advantage and make sure we're providing real world feedback to help enable development of these technologies.
So if I could scratch at that a little bit more, with the advancements in instrumentation, some of which you might have seen here, aimed at really closing the manufacturing process, what are the most important attributes that you see or considerations that we need to address as developers of instrumentation in order to advance the thinking in the field? I'll open it up. Yeah.
Sure. I think one of the things is, for example, connecting to a Delta V system, right, where you can then collect information and going towards sort of automated workflow. I think that's some of the instrumentations today that some of the vendors don't have that, and you can get into the early phase study with that fairly manual, but without that digital connection, it's hard to scale up and standardize. So I think that's maybe one thing instrumentation vendors could do, and I know you guys are certainly doing that. So that's definitely one thing, and that Genentech would be looking for any sort of equipment. Besides the closing, right? And so that's something. And we already talked about the supply chain, and I think that is so critical in terms of your material attributes that you want to develop. And maybe we'll talk about that a little bit more as we get into testing. But
it's a great segue into the next theme, which is around controlling critical process parameters through digital integration. And I certainly think Delta V is one of the predominant ones in the industry. And again, drawing from the biologics history with manufacturing. So I guess as new breakthrough technologies and instruments are being introduced into manufacturing, what are the technology complexities that are going to prevent us from moving towards standardization?
I have to say, I'm 6 months into my new job, and we're an autologous cell therapy company. And one of the things that's interesting to me, you go into our process development lab, and you'll see the exact process equipment, everything that we're running in our manufacturing suite. And in some ways, that's a really good thing. And then you realize that, the industry's come a long way, and it's continuing to progress. But even things like being compliant with the regulations around Part 11 and just simple things like having unique logons for individuals that are using the manufacturing equipment, which today is process development equipment, but tomorrow will be GMP manufacturing equipment. Those are, I see the suppliers, they're, when you talk to them, they're completely, aligned and understand where we need to go. And at the same time, we're not there completely. If you talk about industrializing, it's really understanding those GMP requirements, and companies like Kite are crossing that bridge and helping to kind of pave the way. And there's still enough, I would say heterogeneity or even options out there for technologies where, as we discussed, I think where we have a ways to go to make sure we can translate what we're doing in the labs to something that we can do over and over again, and hopefully in a reproducible and automated way.
When you think about automation, does your mind go more to islands of automation, so components that are fully automated and digitized, or do you think about the end-to-end process of automation as occurring kind of in one fell swoop? Is it something that you think is going to be happening, I won't say in a piecemeal, but in a more modular fashion?
I think depends on the technology, right, that you're developing. I think there's a lot of new technologies coming in from the research area. Gene editing is a really good example. Things are really happening in that area. So today it may be hard to say one end-to-end process will work because our researchers are doing something different, right? They're inventing essentially the new technologies that we will be using in manufacturing, process development manufacturing. So we have to go, at Genentech at least, we always go with the science, right? What the scientists tell us, what are our basic understanding of the biology, and then translating that into a robust process. So in my opinion, I think it's the upfront research technology are changing so fast. There's so many changes and new things coming in that we sort of have to, it's hard for me to imagine a process that will work for the whole workflow. But that being said, it's very important to develop your process from end to end. If I may add a little bit more about digitalization, it's not just digitalization of your process, but especially for autologous, that data needs to sit under, I think a patient management system, where when the doctor orders a product, to that order gets sent to your manufacturing facility, to the back end, the real-time release, because we have to turn these products around. I mean, I think Kite holds a record, in terms of turnaround time, right? That is so important to our patients and doctors. And then the back end, shipping that out, we have companies like Venetti and whatnot that's doing that integration. So I think digitalization is not, for this group, not just looking at the manufacturing piece, but it's testing, it's upfront orchestration, right, of the entire, from the ordering to the administration of the product. I think we have to think holistically about that.
Yeah, and I certainly think that if we ever get to a place where we can accomplish adaptive manufacturing, I mean, this is going to be critical in terms of the digital integration. Sophia, I've heard you talk a couple times about the secret sauce that Kite holds for manufacturing, and I'm wondering if you could comment a little bit more on that.
Well, if I commented, they would shoot me. But what I would say is, to Andy's point, you can't look at manufacturing as a individual unit. And I think Andy made a very good point about, we call it digital vein to vein. The intricacy of the interactions with the, what we call treatment centers, the institutions, and how that information flows into your systems, which often could be, whether it's manufacturing systems, but there's all these data points that are coming in, and the fluidness of that information flowing through as your manufacturing becomes part of your manufacturing. So I do think that is, if the industry is going to move forward, that's the pivot from biologics, is that you can't get away from this whole interaction with the medical community, the institutions. And whether you have case managers or however you're doing that, there needs to be a holistic view of that, coupled with your manufacturing systems. And some of the modules and consumable, the single-use technologies are coming in, they're starting to have their own systems that overlay, and how will those communicate with your systems? I think that's something that we're all starting to look at differently. There are some of them that are bringing some very sophisticated communication systems for their modules, and how will they feed into your manufacturing systems, and as well as in your overall digital vein-to-vein system.
Thank you.
And where's the truth? Where's the data going to reside, and how do you deal with health providers and regulatory agencies around that information?
So Jerry, drawing on your 32 years at Genentech, and now that you have, kind of a new start, so to speak, I'm going to ask you the magic wand question, right? So if you could kind of design right now what you wanted, and you could execute that at Graphite, what would that look like?
Yeah, I have to say I'm very myopic these days. We're, as I said earlier, we're about to enroll our first patient, and we're an autologous company, so at least for now, and so I'm very focused on making sure that everything is smooth, and that our early stage studies get off the ground. So it's really fun to hear about and also read and learn about what Kite's done over the years to industrialize our cell therapy manufacturing. In terms of magic wands, I mean, there's I'm just imagining, I've been out talking to suppliers and we don't own our own manufacturing facilities. There was a really great session yesterday on make versus buy. And I think being able to imagine, scaling out, and our lead programs for sickle cell, again, another great session yesterday on sickle cell disease and where we are in terms of treating sickle cell, and there's certainly other companies in this space that are further ahead than Graphite, and at the same time, it's right now all of the therapies are autologous in nature, and therefore imagining expanding out to treat patients around the world with this imminently treatable and hopefully curable disease. The magic wand there is having scalable solutions and thinking about, what that factory will look like, that industrialized factory will look like, not just the factory, but the control systems behind it. To, if we think about this session and what is industrial, what are the features or characteristics of industrializing our industry? It's, it's at the end of the day, it's access and creating access for all of our, important and advanced medicines, and we want to do that for everyone, equitable access. And so how do we do that and make sure that our treatments are both very effective, uncompromising in terms of safety and quality, but also cost effective? And so the magic wand kind of hits all three of those, quality, cost, speed, and things. And I don't know that there's a magic wand, but there's certainly a lot of great minds that are, in this room, in this conference, and out in the industry that, what gives me hope is that over those 32 years is looking back and thinking about all of, we had to solve a lot of the same challenges and questions. It's like, hey, can you even characterize a biologic? Those early days, those are questions. And then, because if you can't characterize what you're making, then how do you ever hope to make an improvement? And I know for our company, it's early days, but we already have what I'm calling a version 1.1, and we haven't even, treated our first patient yet, but we know we're going to get better over time. So all of those challenges, in terms of that, I'll call it a tension between, hey, we need to make sure that we're being very thoughtful in anything we do and changing and improving our process, to avoid any unintended consequences, comparability challenges, things like that. And at the same time, we want to advance our industry and do the very best we can. So, yeah, so if you have a magic wand for all that, that'd be good.
I do have a magic wand looking back at the antibody space, right? And that was the idea of a well-characterized biologics,
Yeah.
right? Which is what really made the difference for the antibodies field. We know so much about antibodies, and we can make process changes, we can change sites, change raw materials, but still generate a product that's safe and efficacious, right? And that's all based on the understanding of the critical quality attributes of the product. So I think if I had a magic wand, I would say cell therapy, gene therapy become well characterized, and that's really that is an industrialization sort of objective, right? That we shouldn't overlook. I realize the tools are not there today, but maybe we'll look at characterization a little bit more on setting you up, and so that those characterization tools, next generation sequencing, single cell sequencing, if you think about it, that's like mass spec, right, in the biologics protein area. And so what are those tools that as an industry we need to develop and work with the regulators and, to really understand critical attributes and share that data among us, right? And kind of, if you think about the well-characterized biologic CAS meeting, it's everybody shares their information, right? And the whole field moves forward.
And can I add one comment to what Annie said? Because I think it triggered something else that you're seeing in the industry. Because of this effort to characterize, in the cell therapy space, we're starting to allow partners in upstream, and we're not handing them, once we have our processes characterized, then we say, here's our equipment and our manufacturing that we're going to, the manufacturing equipment we're going to use for bioprocessing equipment. We're letting them in earlier so we can partner around the development of how to then, scale that. And I think that is the secret sauce. If you want to, you've got, and then that's being willing to share and co-develop what it will take to scale that. And that's I will share one of our secret sauces. I think, and you probably have heard of our, it was publicly announced, we partner with Draper to help us do that as well. And we're also in relationships with another company that I don't know how public that is helping us, but letting them in further upstream, not waiting and then handing it over. Because if they're co-developing, and not being afraid that with that co-development relationship, at the end of the day, it's going to help the whole industry, because once we get to a certain stage, then they can commercialize that from a manufacturing perspective and make that available to more cell and gene therapy companies, which is how we're working today.
So maybe carrying through on the analytics theme, thank you very much. Jerry, do you want to maybe comment on how analytics for cell therapies may differ in substantial ways from how we think about analytics for other modalities?
Yeah, I mean, there's people in this room that know far more than I do on the topic, but I can say that, for most of us, probably we're working on things to understand the mechanism or mechanisms of action for these complex therapies, and being able to first understand the mechanism of action, then second translate it into something that's a biologically relevant method that you can run reproducibly in a QC setting it's certainly a challenge. And yet, I mean, companies have done it, and we're certainly, down the road. But some of the technologies that we would use for, for biotech are, less relevant, let's say, or at least relevant perhaps for a cell culture, but things like cytometry and things take on a whole new purpose when you're talking about a cell therapy company in terms of characterizing, again, the mechanism of action of your products. And then, of course, as you mentioned, Andy, things like single cell or next-gen sequencing, analytical techniques are, very much, you can imagine, it's early days, but you can certainly imagine these things being critical components to a control system for a cell and gene product. So, yeah, so there's, just the overall complexity. And then, we haven't talked yet about, our biggest source of what I'll call our biggest source of variability, which is especially for autologous programs, which is the patients that we're serving, and, just the various genotypes, phenotypes that we are treating, and just understanding that is in and of itself a pretty big analytical challenge. And, it's kind of forging new partnerships, relationships in the whole paradigm of developing medicines. So for me, it's kind of fun. We have a translational science lead, and she's interviewing candidates for our analytical leads at Graphite, and I think that's probably the case in many other cell and gene therapy companies. And I think it's completely appropriate, and it's a great thing. It's fun, and critical.
So as you just mentioned, Jerry, the quality and quantity of patient cells is really, variable and unpredictable, right? And so how are you thinking about improving the yield on manufacturing processes consistently? Maybe we talked about in-process analytics a little bit earlier, but other analytical methods that might be important in the yield consideration.
Yeah, I mean, there's, we're working right now with hematopoietic stem cells, so there's not as, obviously the field's advanced quite a bit in CAR-Ts in terms of understanding the characteristics, how to cultivate these cells, how to achieve, predictable cultivation. With HSCs, there's different challenges, but what I'll say is, from when you take a step back, at the end of the day, having a robust process, it starts with, your process design, no big secret. For us, what I'm grateful for is that, we're starting with patients that have been apheresed, but we do our selection of our stem cells, and we can freeze that step. So we can be sure that when we start our gene editing process, that we have the right number of cells, and that our gene correction and our cell yields downstream of that are so far predictable. But it's early days for our company for sure. So I'm here drinking it in, so to speak, taking it in and learning. But I'll say, it kind of starts with your process design and being mindful of those things and trying to really design in, again, especially for autologous programs where you can measure at certain points, but for us, we're ex vivo, so we can measure probably, obviously more things. before we treat a patient. So we're very mindful of that as we design our process.
Makes
Yeah,
sense.
if I might be just comment on the yield question, right, or dosage, right?
Yeah.
I mean, I think for biologics, we have fairly predictable PK, PD studies that you could do. You know how to dose. But here, we're talking about cell persistence. How do we measure that? How do you measure efficacy in that context? And what's the right dose, right? Very often we're making manufacturable dosages. It's hard to reach an MTD for a lot of these cell therapy products. So how do you connect that back to a dosage that during your development, right? And I think a lot of the early CAR-T programs have looked back on their dosages, and established that efficacious dose. So I think that's an important part of industrialization, to kind of plan that early, so that you're working with your pharmacology team, clinical team, to really establish the right dose. I think dosage question will also come in with very disruptive technologies. There are companies that's trying to make their T cells, for example, more fit, for example, right, and persist longer, or really generating PD-1 knockout and things like that. So there will be other innovation that's part of the dosage question that we need to pay attention to as an industry, right? And where are they going? What disruptive technology? Because I think about antibodies, right? We used to make antibody, we used to make antibody in the hundreds of milligrams per liter, right? In the early days. Now we're making 10s and 15 grams per liter, right? And that took decades, right? So, and there are disruptive technologies along the way that, as process development manufacturers, we need to pay attention to those changes, because it's coming, right? So we can anticipate science, right, that will transform how we do things.
Yeah, maybe just to build on that briefly, Andy, reflecting as well, if you ask the question, gee, what led to those, yield improvements, with cell engineering and things like media formulations, where we went from very undefined media, didn't know, the secret sauce was not really known, and that's why it was a secret. But now we've got much more defined media in that space, in biologics, and also, we're able to engineer cells using targeted integration and other things to really amp up the output. And you can imagine for cell therapies, a lot of those same solutions are out there. Yeah.
So looking at that a little bit more deeply, when you think about analytics for cell therapy specifically, what's the compare contrast with the analytics that you would apply to biologics? And you talked a little bit about CQAs and those considerations, so they're going to differ, right? So wondering if you can comment on that.
Yeah, I think the key thing, at least for autologous, right, is this chain of custody, chain of identity. Of course, there's a control around that, right? But there's also the real issue, turnaround time, right? I think what are the, we really need real-time release, right? Because patients are waiting. So what are the analytical tests that's really required to give you very, in time, almost just in time, every batch, right? So things like BAC-T, right, or micro-PCR, for example. Today, they're about 7 days. As an industry, can we do better? Can we develop next-gen sequencing method that can give us answers much quicker, right, in hours, for example, right? And we do flow assays, lots of that, but potency assays still takes days to do. You have to thaw cells, target cells, and whatnot. So can we do better at really reducing those times using new technology?
And multiplexing.
Multiplexing. I mean, flow cytometry is great, but there are definitely other systems that we need to think about. And looking at the mAb industry also, we're moving to multi-attribute assays these days, right? And that's here already, right? And also online, Raman spectroscopy for inline measurements, and it's really a lot. I mean, we're sitting on lots of data, right? And for example, Raman, you need lots of data to kind of teach your machine learning, but that's something we could do as an industry too, right? Because that's really going to help. Those tools, right, are really going to help us be much more efficient and make decisions in real time.
Yes. Let's pivot to your favorite topic, Sophia. And we talked a little bit about the focus for graphite and autologous, at least for the time being, but we know that autologous requires more complex, patient-specific supply chain. So in a zero-failure environment, question is, what are the major gaps in supply chain that, we're going to need to be addressed in order to attack the cell therapy market?
Well, I think patients are waiting, to Andy's point, and for our patients, we're right now in the fourth line of treatment, and so, we are always looking at what we call velocity, control speed, and how can we re-engineer their cells and get those back to them super quick, right? Because they're usually in ICU, and they're waiting us for their last hope, them and their families. And so what we learned, and I know you're going to ask this question later, but COVID made us realize that the logistics, you think about, we have over 200 treatment centers, and how are you going to get those re-engineered life-saving treatments back to those patients? And most of us saw a lot of planes not in the air. We mostly use in this industry commercial airlines, and so traditional logistics for biotech, you've got time, but you don't have time. And so when planes were not running or flying, and there was a reduction in flight lanes or paths, how do you get those treatments back to those patients? And so we've got to become a lot more creative around just the logistics of moving these cells, and they're also in cryo temperature containers. And so, how do you do that more efficiently, and then also make it, I think we need standardization for the treatment centers, because there's probably over 200 to 300 clinical manufacturers right now, and then you've got 3 of us in the commercial realm. And these treatment centers are having to deal with all these different protocols on data loggers and LN2s and collection and returns. And I think if we could help the treatment centers with some standardization of some sort, if we came together as an industry, that would help them, and we could also retrieve and return, and then we've got to figure out what we're going to do with the logistics space. I think those were kind of ahas during the COVID period for us, especially the logistics. And then just quite honestly, with warp speed and other things that happen in the supply chain space in general, we have to now think about the external threats a little bit differently, especially with autologous, because the patients are waiting and there's no safety stock. If you have any glitches in your supply chain, that patient may not get the treatment with the hope that they're waiting on. So don't know the answers. We're looking at those. We put together a control tower, and we're working through, and we have a well-established risk. Most companies have a risk management group of some sort, and it's usually business continuity, but we have moved to a control tower where you're always looking at all risk and trying to get ahead of those before they happen. And what will we use to have that information moving forward? I know there's a couple of companies out there that gather data and information, but how will we stay ahead of it? Because we cannot have an interruption in the supply chain.
That's right. Jerry, you were hired during COVID, right? So as you came in with kind of fresh eyes into the graphite, what kind of gaps did you immediately see that were kind of induced by COVID?
Yeah, I went from a very large company which had a lot of leverage with suppliers and things to the spec that no one's ever heard of.
Exactly.
And so yeah, it's still a challenge for us to even get things like pipette tips, and we did rely on the kindness of strangers. It's kind of fun being in this new environment where, we're one of, 6 or 7 companies in this mega building, and we couldn't get some of the basic supplies. Some others have alluded to it here. It's no big surprise, but when you're a small company, it's certainly challenging to, because you just don't have that leverage. So so we have, in fact, when we had all of us got to sign up to meet with, the speed dating format, which is great. So I had a chance to really meet face-to-face here with a lot of our suppliers, and a lot of the conversations we were having were talking, forward-looking, how is our supply challenges gonna, how are we looking for meeting some of the needs, especially for our company as we're entering the clinic. So yeah, it's definitely, still, I would say we're still in supply challenges for some basic materials, and it only takes one link in the chain to cause problems. And as you were saying, Sophia, I mean, definitely transportation lanes, things like that. They're starting to come back, but then we're even noticing with some of our our equipment, our lab and manufacturing equipment, certain processors are delayed, and that we can't get one component for them to finish manufacturing our equipment. So it's it's a tenuous situation.
And I would say it's not even about whether you have leverage or not. I think it's impacting everyone, right?
Yeah.
It's just like chips for cars. If you're looking for a car right now, like I am, there's very few cars on the market because the chips are not available. So think about the sub-tier suppliers for resin. I mean, we had the sterilizers, right, that they had to focus on, the COVID, sterilizing supplies for COVID. And so you couldn't get our suppliers couldn't get their materials sterilized, right? And how do you do that? Do you have letters that, if we came together as an industry, I think cell and gene therapy is unique enough that, and I know the pharmaceutical industry or biotech did write letters so that we could sort of circumvent some of the warp speed halt on manufacturing, and we partnered with our suppliers, so they couldn't get it. So we would give them a letter that allowed them to show the importance of what they were doing for us with our patients waiting. We just have to become more creative. It's not a us, them, and how do we partner together to kind of navigate this, some of these challenges in the supply chain, for autologous especially. Allogeneic, it becomes a little bit different because you make to stock, but you still have an HLA typing matching scenario. So I did spend a few years in allogeneic. I didn't mention that in my introduction. And we were in clinical manufacturing. But, when we had weather interruptions and things of that nature, there was a little bit more breathing room to come up with solutions than in the autologous space. So partnership, but size really doesn't matter with what we experienced with the COVID situation.
Yeah, I totally agree, Sophia, with that. Even at Genentech, we're managing suppliers, not managing, but managing our supplies on a daily basis throughout the whole pandemic, right? Even for us, you think, well, we have leverages. Well, we're just like you guys, right? And everyone's kind of in it together, trying to manage, where am I going to get this filter from, right? Because we file in our BLA this type of filter, right?
That's
well, what do? Do you have alternatives, right? That's the other strategy, right? Is alternative material suppliers. I think a lot of us single source raw materials, right? And that's a huge risk sometimes, right? And I realize early companies probably don't want to think about that too much. You're trying to go fast, but last year and a half really kind of exposed that, at least for us on my programs here at the Genentech T-cell program. We experienced some of that, right? So I think supplier really work closely with your suppliers, making sure they have the right quality system and so on. And partner with them, right, in either scaling up or design better analytical control strategies, get in early, because those things can really hit you, yeah.
So you identified some supply chain risks, and, Sophia, just from your point of view, just wondering if there's some more commonly overlooked supply chain risks that you encounter on a regular basis that you could share?
Andy's just doing such a wonderful job. He needs to come to supply chain. I think he hit the nail on the head. I think we're all rushing to get to market so fast that we're using research-only materials or we're single sourcing. Some cases we have to sole source because there's no alternatives, right? But I think we have to identify those critical materials and move more quickly to put in an alternate or co-develop or work with someone for sometimes a replacement, because we're moving so fast. We've settled on some materials that serve the purpose, but we need to move to something else. And so I think we need to balance that speed. We're now calling it controlled speed, velocity, because I don't know, and maybe in some critical materials, think about adding an alternate up front, or already going from RUO to pharma grade, because when you get to commercialization, the requirements are going to say, You can't have certain impurities, and some of the materials that are research grade will have impurities, and now you got to scramble to change, and I'm living some of those, and a lot of us do live those challenges, and how quickly can you get those addressed is then added burden that maybe could have been avoided if maybe before you get to, it's okay in early development, clinical 1, 2, but maybe when you get to clinical 3, before we go commercial, we should start looking at our materials a little bit differently just to mitigate some of the risk. So I think we're all living with that. I've lived with it pretty much every therapy or modality, but I think it becomes more critical in the autologous space.
Okay, so we don't have fun audience participation, polling, or anything like I saw some of the other sessions do, but we would like to open it up to the audience to submit some questions, and so I think there's a rolling microphone somewhere within the room, so happy to take questions for the panel.
And if it makes you feel better, with the glare, we can't really see your faces. You're just faces out there, but we keep with bodies, but no faces because the glare lights.
Yeah, I also would love it if you can state your name and your company. That would be terrific for us, so we can kind of put it in context. Thanks.
Hi, can you expand a little bit about the particulates? You actually said no particulates, and that is a huge bar. And certainly the regulations allow you for some. So tell me about that and how are you avoiding it? That's pretty challenging. That's for Sophia, of course.
Yeah, we're all looking at her.
USP 90, it really is asking for you to strive to have no particulates. But as you say, with AQLs, you're going to have some, right? And I think, it's a foreign, let's be clear too, foreign matter. Because there are some medias and that will have some level of particulates. But that is our goal, right? Because when you're dealing with autologous and you're dealing with, bags that are really coming off the manufacturing line, going into a cassette, into going back to the treatment center, if that treatment center sees any visible particulates, they're going to reject it, and then that patient's life is not going to be saved. So we're striving for 0 particulates. It is a goal, right? It's an evolution to get there, and so partnering even with our suppliers, many of them have been, whoa, like this is a lot, especially if they're medical device. And so it's a process, and you partner with them to get there. Will we ever be 0? I don't think so, but it has to be a goal because in the space that we're in now, there's just, it will be rejected, and then that patient's life is not going to be saved or potentially, helped. And we do that in our manufacturing. You don't want to get through your manufacturing process and find particulates because you can't reject, when you reject the batch, that patient, unless you've got 2 bags of collection of apheresis, you've basically, have not given that patient hope. So we really strive to have, and you have filters and you do other things in your manufacturing processes, obviously, to also reduce the particulates, especially before final product. But that is our goal, and if others may interpret USP 790 differently, but we're taking a really stringent kind of view of what that means today.
Yeah, it's probably also important to understand the particulate nature of the particulates, right? There you have intrinsic, inherent, intrinsic, extrinsic particulates, and you are allowed some, right? Because what do you do with intrinsic particulates or inherent particulates? There's actually a great paper that was published maybe a few years ago that talks about this kind of framework and kind of viewing that. So I think it's also important to characterize and build a library of what your particular are in your process in a particular facility, right? Every facility is going to have a different risk factor too. Depends on the process in the facility. So I think it's important for companies to set up a program to monitor their particulates, to characterize them, to understand them, right? So I think it also goes back to control system, right?
Mm-hmm.
Hi, great insights. Thank you for that. My name is Rupa Paik. I work for Thermo Fisher Scientific. And my question was, Understanding the context of standardization to get to the industrialization that we are talking about. And this is a question for anyone on the panel. Living Therapeutics lives and dies by their IP. That's not a pun, but it's everybody has their own IP, and they start a company related to that. Could be microdosing strategies, could be shorter turnaround time, 5 days expansion, any of those, dual targeting of T cells. How can all of these varied, diverse IPs that govern the manufacturing workflow be standardized eventually so that they can have the discipline that biologics and monoclonal antibodies have? Thank you.
Well, I mean, I'm happy to provide a perspective there because it is a really important question. And at the end of the day, I mean, in terms of every company has to ask themselves, what are those things we absolutely need to protect, you know? And at the same time, you know, when you walk into a manufacturing facility, a cell manufacturing facility, I would challenge anyone to point out, you know, gee, that's the secret sauce, you know? A lot of, even for biologics today, if you ask the question, you know, what are those things that are either covered by IP or whatever, but we're still sending programs all over the world, it's those things that you can't necessarily see, you know, the cell engineering and those kind of things. And I would put forward that there's a lot of space, you know, in terms of the things we absolutely want to protect as individual companies, entities, and what we can standardize as an industry. So there's plenty of opportunity there for, you know, that element, which is an important feature. If we're ever going to call ourselves industrialized, we definitely have to have a high degree of, I'll call it platform standardization. And, you know, they may not be identical from, you know, either modality or treatment A and B, but, you know, within a modality, I definitely see a lot of space there. For example, you know, we see already you talk to some of it's great, you know, a great source for opportunities for standardization is talking to some of our CDMO partners who see many customers. And when you ask, you know, when I ask and talk to some of the folks who are practitioners of these cell and gene therapy processes on the manufacturing floor, hey, you know, where do you see opportunities for either improvements or standardization? You know, they'll point to things like some of the setups and bags and lines that, you know, frankly aren't, you know, they're different because companies validated them differently, not because they are anything intrinsic to the, you know, kind of the secret sauce of that process. So likewise in the methods area, you know, analytical methods, you know, there's probably a number in this room who have transferred analytical methods, and, you know, there's definitely, if you're a CDMO with multiple customers, and you have like just enough different in the method to be annoying, then, gee, we have to ask ourselves, why is that? You know, is there things we can standardize? And in many cases, maybe no. The answer is no. But I think asking the question is kind of where it starts. I don't know, other perspectives.
Maybe I just add to that, like, you know, we need to maybe as industry agree on the non-competing areas, you know, like apheresis procedures at sites, and how do we deal with, how do we work with sites, right? I mean, sites are tired of us telling them what to do, right? And so maybe that's a non-compete area we need to think about, right? Are there other non-competing areas of industry we can agree on? I mean, I think one of the things we've done in biologics for years is these consulting companies will come in, like Boston Consulting or Deloitte, they will come in and do industry-wide surveys and understand, you know, what are you guys doing without giving out a lot of details, right? Mm-hmm. Those benchmarkings are really important for the industry to become more mature, if you will, and so we can learn from each other, right? What are the best practices? Because there are non-competing areas where we can agree on, but at the end of the day, that's going to benefit our patients, right? And may have wider access to these therapeutics.
Can you give an example of like a couple of these non-competing areas?
Yeah, I think, you know, like turnaround time, you know, platform processes, you know, for example, for the mAb area, that's, you know, it's, you know, there's lots of that in the literature, how the industry went from, you know, early days to now very platform type of processes. So I think that's one area from the biologics area. The other one is analytical methods, right? Mm-hmm. And, you know, you could have water system for an HPLC or Agilent system, right? And I think there are differences, and analytical scientists would debate, and but I think at the end of the day, there also need to be standardization on those workflows.
Maybe just to build on your point about apheresis centers and things, I mean, I'm aware, as I joined this company 6 months ago, there are working groups out there to standardize things like, all of the supply elements that are intrinsic to apheresis centers. And as you pointed out, yeah, there's a certain level of fatigue there because every company comes in with their own unique requirements that aren't necessarily, they're not necessary. So I'm aware of those things that are out there, and I'm certainly, we don't want to invent anything at Graphite that doesn't need to be invented. Yeah. And if we can, jump on board to what others who are further ahead in the journey are, we're definitely, that's part of our philosophy. And the other area is another specific area, things like freezing, cryopreservation. It kind of gets back to the supply chain. And, we've had some, I'll call it some troubles with, because we selected something we thought was industry standard, then you get out there and it's like, oh, this company has to work with like 5 different things, 5 different shippers and all this stuff, and not necessarily because the process requires it, so.
Hi.
Oh, go ahead.
Sorry. Trey Slumsky from Meisner Filtration Products. I think the panel made a really good use of its magic wand in talking about, trying to establish well-characterized processes. And, that was certainly a breakthrough, I think, as we saw the propagation of MAB processing and everything else. Having spent a decent amount of time with vaccines, which maybe had a head start, but some can argue, but never really became well-characterized processes in a lot of cases. There's a lot of similarities there, I think, with some of these processes. Are there any particular hurdles you see, maybe learning the lessons from vaccine evolution, that could be applied to try and make the magic wand a reality and turn these into well-characterized processes?
I can try. Yeah, obviously a lot of the vaccines, especially some of the more legacy type of vaccines, right, are not considered well characterized. But if you think about mRNA vaccines today, right, that's very well characterized, right? And of course, it's a little bit easier to do that. And so part of it is developing analytical methods that's capable, right, to measure the attributes that you think are important, right? So, and obviously for some of his, more legacy vaccines, they're just more complex, right? So it's very difficult to characterize. But I think, so you kind of have to go hand in hand with your analytical methods, right, and your understanding of the product, and marrying those two together. And I think just because something is complex shouldn't stop us as an industry to strive for better characterization and better understanding. So I actually would challenge all of us, right? Not just because we just can't sit behind this. We have to rise up to the challenge of characterizing and understanding our product. I think that's our patient need it. Our patients, they require us to do this, right? So I think it's a general call to the industry to really work on characterization and there are tools and develop new tools, right, that would be able to characterize our product. Yeah. I don't have a easy solution for you, but I think
that's why you need the magic wand.
Yeah. Yeah.
I'm Kate Dyer. I'm the CEO for StemExpress. We have standalone cell collection centers. And I'm just curious, specifically to Sophia, but to the whole group, when you talk about the treatment centers having exhaustion from just all of the steps that they have to take, and then especially when you talked about logistics in COVID, beating up a lot of not only our own companies, but the hospitals as well, I'm just curious from the starting material standpoint, be it apheresis, et cetera, do you feel like there is a need for sort of large-scale centers to be available to do these type of collections outside of just the standard treatment centers that exist today?
I'll start with, I know there are consortiums and organizations are starting to have those conversations like FACT and some of the well-known apheresis organizations or companies out there. And, you know, I think it's going to depend on what your therapy is treating and how well that'll be successful. For what we're doing right now, the patients are, you know, fourth line indication. They're at a point where they have, you know, very limited time given. And so how do you collect and where you collect from is not like outpatient oftentimes. So it depends on your therapy because there are opportunities, I think, for us to look at depending on the therapy and whether that patient is movable to do the apheresis. But I think it depends on your therapy on what we'll be able to look at. But I assume with 200 plus clinical manufacturers and so many of us in this cell space that there has to be an opportunity there to look at a different model around apheresis collection. And I know others are starting to embark on that. I'm just not sure if we're looking at that as an opportunity to standardize or work together with best practices to come up with what that might look like in the future. But for our current line of treatment, you know, many of our patients are not movable. So you have to go into the hospital.
But I think as these therapies move more towards frontline settings, you will have, you know, or solid cancers where you're gonna, you know, these patients are typically treated in community settings, right? How we collect their cells, right? You don't want them to go to an academic center, right, or center of excellence to collect cells. So I think that's a problem we still need to solve. Yeah, I mean, I think you guys are working on B-cell lymphoma, right? I mean, these are third, fourth line patients. I mean, they're usually seen at, you know, hospital, I mean, pretty academic center with, you know, established, you know, cell processing capabilities. But I think, but it's a really good question. We have to kind of solve that as a field. Yeah.
Hi, Namrita Ravinder from Thermo Fisher. My question is more around information and data sharing. Andy, I think you touched upon it earlier. This wealth of information different industry leaders have. How do we take that whole process from either patient variability, sample characterization, to inline, to residual assays, to release QC, or regulatory for that perspective? All this information, each industry or each industry leader is learning with each therapy new things. How do we mobilize that data sharing, information sharing, so that you can get that speed to that, you know, last line treatment for the patients? There is some competition there in the field, but how do we get that data sharing? So what are your thoughts on how do you see the industry growing in that, coming together as a community and sharing this information? Because, you know, same thing applies for us as an industry suppliers for a lot of these reagents or platforms.
Yeah.
Learnings from the regulatory side. There's a lot of sharing that can be done amongst us within the community. So how do you see this being mobilized?
Yeah,
That's where I see speed is, you know, stuck because a lot of information is stuck in silos.
Yeah. Well, I think certainly, you know, some certain critical attributes are specific to your therapy, right? I mean, a T cell or CD34 cells are not going to have the same CQA, obviously, right? The mechanisms are different. But where we can agree on, right? I mean, I look to organization like CAS, right, for biologics, where there's lots of sharing. And this CAS actually now has a cell and gene therapy program yearly. I think that's a really great start for sharing things like control system, you know, characterization, regulatory information. So I think that's a really good start. And then PDA, of course, has their chapters and so on. I think those are the venues that our scientists and engineers are very comfortable sharing, right? Those are set up. So I would definitely encourage the field to gather and sort of support those initiatives, and also, so I think that's the way I see it, you know, is that we have to use some of the structures that are established already for sharing information. So that's something I encourage everybody to lean into here.
Thank you. My name is Dalip Sethi from Terumo. So one of the question I have, I think, follows the question. When you talk about data sharing, the next question comes to the AI, right?
I'm sorry, what's
Artificial intelligence, AI. But when you need to think about AI or machine learning, you need a lot of data and you need a lot of outcome. So how do you see cell therapy going into that space where the patient data is currently limited or it's in pockets, right? Or the clinical trials are not that huge yet. So how does the industry goes learning that and then connecting into AI and machine learning? What does the future looks like? Thank you.
Yeah, I mean, at the patient level, obviously, for the kind of products we're making, it's not large numbers of patients, but just last week I was, talking with a couple of our scientists, and one of them was, we have some new cytometry equipment and some flow equipment, and they're looking at, they're using AI to pick up things in these flow experiments that humans can't, and there you have large populations of, I'll call it data, single point data, and looking for deeper understanding of the process through those technologies. So AI there, especially for cell therapy companies that are using flow, there's a lot of applications, even if you're not treating large numbers of patients, but yet can give you deeper insights into your data. So it's just recent learning for myself.
I think maybe also think more creatively, right, about AI. I mean, this natural language processing that, like batch record reviews, right? I mean, could there be something that could be applied to that, for example, right? But machine learning does take lots of data. Our partner Adaptive they're kind of an interesting, wet lab company and lots of bioinformatics, right? And so I think partnering with somebody who has that deep expertise is really helpful to know how to apply AI and machine learning. And it's true you need tons of data, right, to really be able to do that. And I think some of the analytical methods that we run so frequently, right, there are ways to automate a lot of that. We actually took on a project at Genentech to automate a lot of our flow analysis. People draw gates and things like that. So people do that all the time, right? There's so much data. So I think that's kind of a pretty good start, right, to sort of test out your machine learning capabilities and train your analytical methods to be more automated. So there are definitely areas. Yeah, great question.
Yeah. I don't know if there's, I'm just looking at the timer. I don't know if there's any other questions, but one element of industrialization, we didn't really have it totally baked into the program, but if you ask the question, how will we know we're there? One piece is, we've talked a lot about processes and technologies, but we haven't talked much about people in this session, although I've heard it certainly in other sessions. And I have to say, one of the things, I didn't use this in my magic wand answer, but one of the things that's on my mind a lot these days is, geez, how are we going to scale our industry with the talent that's there? And how do we get a deeper talent pool? And I'm finding, at least in my my travels, my experience in the Bay Area, that we don't have a kind of concerted effort to help develop talent. There's certainly a lot of things happening, but I wouldn't say it's like super coordinated. But I'm curious, since this is a workshop, we can put you guys to work in the last minutes. I'd love to hear, ideas of how we can, collectively solve the, deepen the talent pool, particularly for practitioners of, cell and gene therapies. My mind's mostly on manufacturing, but, whether you get into analytics, other space, supply chain, there's just a dearth out there. Don't know if anybody wants to pick up on that one, but love to learn.
So one of the things that I think when you think about talent management or training more people, especially with automation, like especially Thermo Fisher, Terumo, like we guys, when we do automation, so you have like some machinists and some biologists, right? You need people who understand the biology to develop the processes, but on a second level, you know people who know automation, right? They may not know deeper biology, so we should have more consortiums going to academia and training people on technologies and how to operate. They may not be a PhD in like immunology, but they may be able to do manufacturing in a line pretty sophisticatedly, having all the aseptic techniques, and should be able to have more people in the workforce from that angle. I think that is also a crunch, not just the biologists, but also people who can do the manufacturing. So that is one idea where we should go as an industry.
Good point. Thanks.
Yeah, I think we talked about this a little bit last night, but education, training, that all kind of goes into it. And there are programs that are sprouting up across the country and I guess globally as well in terms of master's degree programs and automated science and, kind of bridging the divide between engineers and scientists really so that they get that cross-training needed to really be successful in cell and gene therapy. So I think we need to do a better job of training and education.
Yeah, I just maybe comment on that. I went to Northwestern University for my graduate school, and I sit on the board now for them for their master's program, right, where 40-some students every year graduated. And I think that's a good way for the industry to give back, right? And I think, like Jerry said, people are getting poached all the time these days, right? Because there's just so many opportunities, and we're finding it difficult to, sometimes, right, to retain our best assets. I think we talked about that, our people. So it's oftentimes something we pay a lot of attention to technical things, but thanks, Jerry, for bringing that up. I think we could do a better job, right, in reaching back to, at a room full of people. A lot of you guys could probably just make a connection with somebody, and that get them excited, right? That's part of it, just getting our messages out there to recruit people and get young people excited about the biotech in general, right?
And I will just add one other opportunity for us that I've been at 3 companies, and I think, including Kite, are doing things like apprenticeships and partnering with universities. I think all of us have rotational programs, and if you don't, that's another good approach, putting a rotational program in your research and development group or your manufacturing group. So we're finding success there, those 2-year rotations, and then they come off and they're slotted into a role. We have apprenticeships in our facility in Maryland's doing with in the analytical space and in the quality space, just to build some competencies there. So partnerships go well, I think, with universities. And then I love the combination that you shared from Terumo of combining, because I have worked with a CDMO in my allogeneic life that where they were based, we talked about this at dinner, they were struggling. There was really no major universities focused. They have biology programs or biochemistry, but they were struggling in their manufacturing space to find cell therapy specialists. And I think that is happening across the country as well.
Thanks.
Okay,
I kind of wanted to speak up actually I'm Alyssa Wade from Thermo Fisher, and not to toot our own horn as Thermo Fisher, but having UCSF and having that collaboration, I think bringing awareness to university students to know what opportunities are out there, because so often you study one area. I'm just a marketing person, but even coming into marketing, I said, okay, what do I want to do for my career? So as you guys were touching on, bringing it into the university space and things like the collaboration center that we're working on, I think that brings awareness to students to know that's even an opportunity. So biology can be so general, but to know about cell and gene therapy and what that actually does, and me even learning about it in my role has been very exciting, and I would have never known about it as a career role in college. So I think that's another way.
Great, thanks.
Thanks, Alyssa. Yeah, you can't think of a more inspiring kind of topic or area to be in, than cell therapy. So it's not a dry kind of hard sell to, young people, I think. Any other questions? Well, if not, please join me in thanking our illustrious panel here. I think it's been great.